Title of my talk, Grain Yield Increase, the Role for Genetic Modification. What a question this is. Of course, it's a yes. I could stop here right away, but don't worry, I'm not running away. I'll try to do my best to answer you the question as to why we need that technology for improving yield. My introductory slide is actually not really necessary because Joachim has paved the path for me already very, very nicely. Apologies, I'm going with a doubling number that you have moderated is probably not as correct as it is so very often cited. But let me take it as a worst case scenario that grain production needs to be increased. I think there is consent in this notion. To what extent is maybe a different question. And you also said that of course there's the population increase. We're adding 75 million to the planet every year. This is the size of Germany, so one Germany, 1 year after the other, that is a problem. We heard already about the competition with animal feed, where it takes many kilograms of grain to make a kilogram of meat. This we all know, and raw materials. And see the picture down there. This is the peninsula of Malaysia, and I'm traveling there once in a while because I'm looking at biodiversity there, and there's many species I cannot find any longer I have to sadly report. And actually Malaysia, from north to south, from east to west, is full of palm oils, and there isn't much else there left. So we all agree we need to intensify agriculture one way or another, and we better do this sustainably under these time constraints until 2050. This is very little time, and that's not at all compatible with the time scale that agricultural research and development goes. It's a slow process, and this is my number one. The time scales are really dramatically short when you ask a plant biologist to quickly come up with a solution. It's decades. We are not talking about years, and plus then there is climate change and the low investments that we notoriously have into agricultural research and development. This is another issue. As compared to other financial support that goes into interventions for other sorts of human suffering, such as HIV research. This is completely incompatible. Also this, I can deal with this very shortly because Joachim has pointed out already, yes, we are doing yield increases year after year, but see the projections, which are the solid lines. I mean, attaining this doubling, we would not reach it. We would not reach it. If you want to double for the yield for like rice or which is in the 40%, we will not be going there, and we don't really have true game-changing solutions in hand that give us a big boost of yield, although there's precedence for success. And now I would need the next slide, please. Doesn't work. Now here we go. Again, there's many angles to boosting yields, farm management. That's one, but the question I'm here for is to ask the question, what can plant science do? And again, there's many angles to this single question as well. We can deal with biotic stresses and abiotic stresses. This refers to the question that has been raised before. And yes, looking at the scientific literature, you cannot imagine how many papers there are. How many molecular detail there is that tells you about the nuts and bolts involved in drought tolerance, in salinity, submergence, you name them, in all sorts of biotic stresses like viruses and so on and so forth, but we don't see really that this has been ever carried on into solutions. We rarely see that. So all of this avoidance of losses sort of agenda is contrasted is by the question as to whether we can make plants that do not avoid loss but can do more than ever under unstressed conditions, and that is normally referred to as potential yield. And as you know, there is precedence for this. So this is Norman Borlaug. We heard already the concept that he has put forward at the time was to develop those semi-dwarf varieties, which, among other issues, just improved so much the harvest index, which is the proportion of a plant that is harvested. And that made a huge difference in agricultural productivity, especially in wheat, especially in rice, and was a breakthrough. So the question we want to ask here is what are scientists today doing? Scientists today, as compared to Norman Borlaug, have wonderful tools in hand. Genomics, transcriptomics, you name them. They have a huge wealth of molecular data in hand. They can, just through observational studies from a single cross, let's say a high yielding on the right and a low yielding parent, they can decipher which are the areas of the chromosome. This is a hypothetical plant with only 2 chromosomes, would then be transported into the subsequent generations, and what are the areas of the genome that we need to bring together to get high yields. So that's fantastic, and on the happy side here is that this is not a GM, and everybody likes this idea. But when I look at all the literature, and there's ample literature on that, on yield issue, and what can we learn from genomics and all the related technologies, I must draw a conclusion in a nutshell that the conclusion from this research is basically, it's very complex. It's so complicated, yield is something that is related to the whole development of a plant. It goes how is germination, what is like, and what is the seedling vigor, then it grows, and there's pests, and there is architecture, and as a matter of fact, and that's probably very, very right, there's many genes involved in something as complex as yield is. So there are many trait associations, as they term it, and there is a dogma out there that is not outspoken so very much, but that I feel is there, that all of my genetics and genomics colleagues say, hey, there is not a single most important gene around that can drive yield up. And it's just too complex. However, I wish to contradict, and I'm going back to next slide. I'm going back to the Green Revolution gene. This is how we call them. This is the semi-dwarf concept of Norman Borlaug as we know today. This entire concept is driven by single most important genes. In rice, seen up there, it is all about the biosynthesis of a very important compound. Is a phytohormone called gibberellin acid that masters elongation. So not producing enough of this hormone makes more stunted plants, and they are high yielding. This is the situation in rice, and in the wheat it's on the same pathway, but it is downstream of this phytohormone. It inhibits the response. So rice cannot make enough gibberellins, and wheat is blind for gibberellin, and those are 2 genes. They have made a huge difference. And if I had the money and the say, and both doesn't apply really, I would always go and welcome, I'm skipping over the slide, excuse me. I would always welcome a research proposal that as an international consortium entitled Single Gene Solutions to Break the Yield Barrier. And that this is not a dream, I want to just give you by details by some examples that are in the recent literature that I know of, and some of which is his own work also. So, for instance, here is my colleague, Professor Sun. He's a Chinese fellow, but he works in Sweden, in Uppsala. And he comes with this issue here, where he transforms a single transcriptional activator called SUSIBA2 under an endosperm-specific promoter control, so it's only active in the grain. And what he finds is a yield increase of plus 30%. Field tested for 3 years in China. So you can see the difference of those tillers, panicles, with those hanging, heavy, much more grain that all fill. They are much more stacked with starch, plus 30%, and that's not all, because as he can consistently show on the next slide, is that methane emission is reduced by 90%. Somebody has calculated, I don't know the basis of this calculation, I'm putting it just here for the show, that it would be equivalent of removing many cars off the road. So see those blue bars, these are the wild type, and the red little bars are the transgenics, and this is age. And 28 days after flowering, there is no more methane emission from those paddies. Isn't that fantastic? And on the right-hand side, you see just the colonization of those roots by methogenic bacteria. And it's all about a redistribution of carbon that goes more in the grain than in the root, and the methogenic bacteria wouldn't colonize those roots any longer. As you see down here, there are no more, practically none of those bacteria. Methane reduction by 90%, a yield increase by 30%. I find this pretty much impressive. This is a bit of own work. We find that by mastering a process, a biological process that is called programmed cell death, or apoptosis, that this limits growth. So by using a single anti-apoptotic gene, one gene expressed only in the grain, gives us much larger grains, as you can see in the middle there on the left panel. This is the wild type. On the right-hand side, you see how large of those grains grow. So 10% larger grains, but in addition, same story. We are getting much longer panicles, many more filled grains, and we have consistently over 4 or 5 years that we are doing this research, plus 30% at average. Then talking this through with my American colleagues like Bob Buchanan and Peggy Lemau, they say, hey, we're having something very similar in sorghum. Isn't that interesting? We can make larger grains with a single grain, and we measure that we have plus 16%. And then here come the Brits, haven't published anything on this, but make a big press releases. But they are very renowned institutions. Rothamsted, University of Essex, Lancers. Go and you can look at this on the internet. Here come yield increases of 20 to 40% that they claim, and that's going into the field as they hope in the next year. What I want to stress here is that those numbers at times where everybody is asking for a consistent yield increase of plus 2 or 3, this is fantastic. It's unprecedented and really, really impressive. However, the problem is that I'm not saying forget about breeding, but breeding, we all know how breeding goes. It will bring fractional increases over time, but we don't have that time. But while those GMs are just game-changing or bear that potential, however, all of these discoveries that I showed here with no claim to completeness, there are others out there, are all in the public sector, and none of those researchers know where to go from here. So they will write a super publication and then turn to a different problem because it's interesting. And my very last slide came to my mind is when I looked at the last Nobel Prize in Literature, is that not taking advantage of options bear the risk of drowning. Thank you very much.